Yes, everybody typed in yes, they can hear you. We're good to go.
Great, great, we're good to go. Okay. Again, folks, sorry about the delay. Welcome to Building Strength webinars. We have quite a good attendance tonight. This is fantastic. As you see, and as you can see, there's a disclaimer up there, and I guess we have to do that now just for legal reasons. But the idea behind these webinars is not to give you any kind of medical advice or consultation or diagnosis or treatment with regards to treatment, but the idea behind these webinars is to really teach you how the body works and what you need to do to keep it working. And that might seem simple enough, but from our experience and from what we've seen, it seems like there is a huge need for the simple, straightforward teaching in that regard. And really, again, this is not to replace your doctor's advice. This really, folks, is an area that most doctors are not really paying attention to. And particularly this class today on cancer, this really, about 2 years when I was in radiation oncology training, it was during that time that really that was when the seeds were planted for what we're doing today. And of course, Building Strength webinars were started about 7 months ago with me and my friend and partner, Sherry Platt, who's on the call. I say hello, Sherry.
Hello.
And the idea was really to help people understand these basic principles. And on the screen right now, you see a title of a DVD actually called Remaking American Medicine. And this is a remarkable DVD. It's really about doctors in their field, well, not just doctors now, but healthcare professionals, all looking at the state of American medicine and deciding to organize, to rally the troops, so to speak, to do something about it, because it is just unacceptable. The rates of mortality and the disease and how people are falling because in this country that has one of the best technologies in the world where medicine is concerned. And in particular, there are about 5 one-hour shows, but one of them in particular was about diabetes. And folks, there was this man, he's a producer, a Hollywood producer, not just any Hollywood producer, but the producer of the movie Platoon and The Fugitive. These are movies that won Oscars. Well, this man had diabetes, and he was closing in on kidney failure and dialysis. He was looking at dialysis until he switched from his doctor to another doctor who could actually educate him, and that was the key word, educate him on what he had to do to ensure that his treatment was working well. And his other doctor was not doing that. Now think about that. Think about that. This is a very intelligent man with almost unlimited resources, and yet, so he could have found any doctor to treat him, and yet he wasn't getting the kind of treatment or the education that was necessary. And so that's what we're doing, and that's why we do what we do. Now, really, we have a very special guest to help us do that. But before I ask Sherry to introduce him, the idea behind, again, what we're doing is your doctor focuses on the right side, the medical approach. So if there's a problem, let's say pain, he focuses on the pain and he tries to deal with the pain. I'm going to show you a slide right now that actually demonstrates that, if this thing will come on. Okay, you might be seeing a mountain that is being attacked with medications, but at the same time there are side effects. And also to the left, the natural approach, the mountain which refers to your health and your healing, that is also being affected. All medications, all treatment can have side effects, adverse side effects on your body. Well, what are we going to do now? If we focus on this is what we're doing, if I can get this thing to work. Come on, folks. Okay, next slide. Okay. If we can focus on health, now you see also an orange, an apple, you see sunshine, you see the tomatoes, sunshine, and there should be a dumbbell there somewhere. Anyway, if you strengthen the human body, if you give your body what it takes, and we talk about the wisdom of the body a lot, if you give your body what it takes, it in turn will help to correct some things. It may not be able to correct everything, but it will go a long way towards making your medication work better. And so the whole idea, I hope Dr. Murray doesn't have the same kind of trouble with that I do and that I'm having. So the whole idea, folks, is let your doctor take care of your disease with education, with knowledge. And that's what the WHO is talking about. There are two obstacles to vibrant health and longevity: ignorance and complacency. And with these webinars, we're trying to deal with the first part especially. So, of course, we focus on three main areas: the power of our body, the wisdom of our body, what our bodies need, that includes nutrition, health, that eventually result in diseases like cancer and diabetes. And so without much further ado, I think I'll just hand over to Sherry. Why don't you go ahead and introduce one of your favorite people to the webinar?
I'm going to go ahead and make him the presenter as well.
Okay.
Is that Dr. Murray?
I got it, yeah.
All right. So Dr. Murray...
Sorry, I don't know if I can... Yeah, I've got the slides going. Okay, sorry.
Okay. We're good, and we see your screen.
It's sharp.
Okay, I'll whittle it down to 10 pages. Okay, Dr. Murray received his medical degree from the University of Glasgow, Scotland in 1956. He of postgraduate training at the University of Michigan in the United States, where he was awarded an MS degree in physiology in 1958. This was followed by 3 more years of graduate research in biochemistry at the University of Toronto, Canada. Upon completion of his PhD, he received an appointment as assistant professor at the University of Toronto. In 1965, he was appointed Senior Postdoctoral Fellow Oncology and Pathology, that is a mouthful, Toronto, in 1968 as Associate Professor in Biochemistry, where he was appointed a professor in 1973. Dr. Murray had a distinguished teaching and research career at the university and trained more than a dozen graduate students. He published over 50 scientific peer-reviewed papers, authored multiple textbook articles, and is one of the authors of Harper's Biochemistry, the 27th edition of which has just been published in 2006. Since 1998, Dr. Murray has served as Professor Emeritus at the University of Toronto and recently as a consultant in carbohydrate biology and biochemistry to the nutritional supplement industry. That's all that we have about Dr. Murray.
That's more than enough.
Well, Dr. Murray, thank you again for agreeing to do this. And we know this will probably be the first in a couple of other webinars if we behave ourselves. That is.
That's true.
Okay.
Okay.
We got the camera.
You want me to go ahead?
Yes, sir. Please go ahead.
And many thanks to David for inviting me. And also, Sherry, I wish to thank her particularly for all the work she did getting the slides ready. And just briefly, I'd like to say hello to my friend Robin Sully and her family in Brisbane, Australia. I believe she's listening. So we're looking at the first slide, and if you have any questions subsequently, you can email them to me, and I shall try to give you as reasonable an answer as I can come up with. So that's my email address. I live just outside Toronto in Canada. Okay. So this is a very valuable reference that I don't know if it's still in the local bookstores, but there was an edition of Scientific American called New Answers for Cancer, okay, volume 18, number 3, 2008. And I found that a particularly up-to-date issue of Scientific American and presenting what I'd call the scientific and medical thoughts and other healthcare workers too on cancer. There may be people with different views about cancer out there, but I'm more or less presenting what I believe is a fairly current scientific and medical view of cancer. And another useful resource is the American Cancer Society website. There are doubtless hundreds of useful websites about cancer. So here are some of the topics to be covered. Okay, some definitions that will be a bit dull. We'll briefly mention some of the causes of cancer. Then we'll address the characteristics of cancer cells, how they differ from normal cells. Then we'll talk a little bit about the spread of cancer cells, the staging of cancer. I'm only going to address some treatment topics fairly briefly, and this is not medical advice. This is just telling you what the standard treatments are and what some new approaches are. And in terms of outlook of cancer, the most important thing I'm going to mention is early diagnosis. Okay, if you think you have anything abnormal, you should see your healthcare practitioner as soon as possible. So here is a brief comment. Cancer is a huge subject. In a short section like this, we can only touch on certain aspects. Hopefully the material covered will provide those of you whose knowledge of cancer is somewhat limited. It will provide a jumping off spot for seeking further information. Now, much has been learned about cancer cells, but much remains to be learned. And not all cancers are identical. There are many different types of cancer, and they have different properties to some extent, and that makes matters more complex in terms of treatment, etc. Okay? So the webinar is not about any specific treatment recommendations. From my perspective, is to give an overview of basic aspects of cancer cells from a scientific and medical viewpoint. Okay, so I won't be talking about various ways of treating cancer very much. I'm going to try to present to you how I believe most physicians think nowadays and scientists of the causes and behavior of cancer cells. So here are some definitions. Cancer is any malignant tumor, and a tumor, an interchangeable term, is neoplasm, which means neo means new growth, a new growth. A tumor is an abnormal growth of tissue. For example, we know breast, prostate, stomach, almost every organ in the body that may be benign or malignant. And as I said, neoplasm means new growth, and in fact, you often refer to this. You often hear the study of cancer referred to as neoplasia. That is a study of new growths. Okay, the science of studying cancer, or also the word oncology, which is a study of cancer. Onkos being the Greek word for growth or tumor. So a benign tumor is a tumor that does not invade the tissue in which it arose, and it does not spread, or the medical word metastasize, to other sites. But a benign tumor can sometimes cause serious problems. For example, if it's in the brain, it's growing in an enclosed space, and that can cause serious brain problems. Or some benign tumors can bleed, or can obstruct the gut, etc. So the word benign refers to the fact, basically, that it does not spread. And a malignant tumor then can invade and destroy the tissue in which it arises, and it may spread or metastasize to other parts of the body. Okay? So carcinoma is another term that many of you may have seen. That's a cancer that arises from epithelial cells. These are the cells that line the skin and the internal organs, for example, the colon, the little tubes, ducts in the breast, the prostate, etc. And a sarcoma is also a cancer, but it arises from connective tissue cells, such as the cells underneath the skin, a variety of cells. So metastasis then is the spread of cancer cells to another part of the body, and the spread may occur via the bloodstream, or it can occur via lymphatics, or it can occur across body cavities. For example, if a tumor arises in the abdomen, let's say the colon, it can spread across the abdomen. Cells can get into the abdomen and spread to elsewhere in the abdomen. So these are the three major routes of spread of cancer cells: via the bloodstream, via the lymphatics, which are little tubules, tubes basically, that eventually drain into the bloodstream or across body cavities. And leukemias are malignant diseases affecting various types of white blood cells. They may be acute, that is, relatively rapidly progressive, or they may be slowly progressive, and they may affect lymphoid cells, which are cells involved in the immune system, or they can affect other types of white cells, and these are often called myeloid cells. So you'll hear the terms acute myeloid leukemia or acute lymphocytic leukemia or chronic lymphatic leukemia or chronic myeloid leukemia. And lymphomas are cancers that arise basically from cells of lymph glands. Some of them may be relatively benign. Okay? Some causes of cancer then. Here we have a cell, and here I've represented the DNA in the cell because the DNA is critical. Our genes are critical in cancer. Nowadays, most people believe that cancer is a disease of the genes. I don't necessarily mean that it's inherited, but in some cases it can be inherited, but it's a disease in which genes are affected and they're changed and they give new properties to the cells, in fact, the properties of being a cancer cell. So regarding causes then, there are hundreds, if not thousands, chemicals that can cause cancer. Most of them interact with DNA. This is the double helix of DNA. Of course, there's 23 pairs of chromosomes in a human cell. I'm only showing a very limited area here, and there's about 26,000 genes in humans. Okay, that was established by the Human Genome Project. So chemicals interact with DNA without going into the chemistry, and they cause a change, a mutation in DNA. Again, I'm not going to give the details of how chemicals cause these mutations, but mutations are a key event in cancer. Similarly, radiation, different types of radiation can affect the structure of DNA and cause mutations. Certain viruses introduce new genetic material into cells. For example, the papillomavirus that's gotten a lot of coverage in the past few years because now many people are recommending a vaccine. And in fact, one of the scientists who did this was a recipient of the Nobel Prize in Medicine this year. Okay? Then there are other what we would call genetic, in a small number of people, for example, some people who are predisposed to breast or ovarian cancers, some other cancers, there are already mutations in their DNA at birth, and these predispose to cancer. So these are the major causes of cancer, hundreds of chemicals, and the common feature, the most important feature, is that most of them affect the structure of DNA. They cause changes in DNA. They cause changes in the structures of important genes, genes that have various properties that I'll talk about in a minute. Okay? So here are some comments on the causes of cancer. Certain chemicals and radiation cause mutations in DNA in certain key genes. There's 26,000 genes, but not all of them are involved in cancer. There might be 100 or more important genes. We still don't know the exact number. Viruses, which can be made up of DNA or RNA, they can introduce new genetic material into human cells, and that interferes with the function of normal genes, and that can tip the cell to becoming a cancer cell, for example, the papillomavirus. And certain individuals inherit mutations in specific genes that predispose to cancer. I guess many of you have heard of women who, after a genetic screen, and they found out that they have a cancer susceptibility gene for, say, cancer of the breast. Some of these women have opted for a mastectomy. I'm not going to discuss that in any detail here. But mutations are basically changes in DNA structure. For example, changes in one unit we call a unit of, the simplest unit of DNA is a base pair. DNA is made up of bases, and these bases pair with each other to give the two strands of DNA. So you can change one base, and that will change the structure of a gene, and that will change its function. In other cases, you can have more extensive changes, deletions, and you can also have fusions of genes. So there's a whole variety of mutations. The majority are the ones that affect one base pair, but there's a variety of other important mutations. And mutations are a key factor, we believe, in many cases of cancer. So what is the result of a mutation? Okay, so a normal gene, most normal genes make proteins, okay? Some genes make small molecule RNA molecules. Maybe out of the 26,000, a couple of thousand do that, but the majority make proteins, and the proteins are the molecules that perform the work of the body, okay? So we're talking about a minimum of 24,000 proteins being made in our body, quite a bit larger than that, and each has a specific function. So if we have a mutation in a gene, it makes an abnormal protein. In some cases, it may not make a protein at all, okay, depending on the mutation, and then the normal function of that protein is not performed. So this can have serious consequences for a cell, rendering it more susceptible to becoming a cancer cell. And there's evidence from many studies that at least 6 key genes, and maybe more, may have to be changed in most circumstances for a cell to become cancerous. I won't go into the experimental detail of that, but for example, the Scientific American article that I mentioned does go into that in quite a bit of detail. So what then are some of the characteristics of cancer cells? Well, I attempt to show this in this slide. Here's the cell, cancer cell. This is the genetic material. This is the membrane surrounding the cell, the surface membrane. Okay, so cancer cells, normal cells usually respond to signals that turn on their growth, okay? Signals might be hormones or growth factors, whereas many cancer cells grow in the absence of normal signals. Their growth is unregulated. They're independent of normal signals. And also, they will ignore other signals that tell normal cells to stop growing. So abnormal growth, as we know, is a key feature of cancer. Growth in the absence of normal signals, where a normal cell would need signals, or growth despite signals that would tell a normal cell to stop growing, okay? Then another interesting phenomenon that I won't have time to go into in great detail, but there's a phenomenon called abnormal cell death. Cells die after a certain period of time, okay, and there's a whole complicated chain of reactions that cause normal cell death. This is given this word apoptosis, which is derived from the Greek. But so apoptosis is basically what we call programmed cell death. That is, a series of reactions occur in a cell turned on by different signals, and these cause the cell to die. But it appears there are abnormalities of apoptosis in many cancer cells so that they do not die as normal cells do, okay? So another interesting phenomenon about cancer is if you look around a cancer cell, you often see a variety of sort of abnormal proliferating blood vessels. And there's a lot of interest in the blood supply of a tumor, because if you could cut off the blood supply of the tumor preferentially without interfering with normal blood supply, you could kill the tumor. And this phenomenon of the growth of new blood vessels is called angiogenesis, okay? That means growth of new blood vessels. And if you take cancer cells and you take them out of the body and put them in a glass dish, tissue culture, and grow them in a chamber, okay, cancer cells, after a while, most normal cells will die, whereas cancer cells can generally go on growing and growing indefinitely in tissue culture. So perhaps the most, sorry, go ahead.
Dr. Murray, sorry to interrupt, but what makes that happen? I mean, how come without nutrients, they just keep growing even without nutrients?
No, I'm talking about nutrients being supplied in the tissue culture, okay? So it's probably got a connection with the programmed cell death, that they no longer have the same programmed cell death that normal cells exhibit, okay? So these two phenomena, immortality and apoptosis, are linked, okay? The understanding of apoptosis and why cells grow forever, this is quite a complex area that hopefully, if you're interested, if a listener is interested, they can take off and look at. There's sites on the web. If you access PubMed, PUBMED, the National Library of Medicine in the United States, if you enter the word apoptosis, you'll find thousands of references. It's a huge area, as is this, which is probably the most serious characteristic of a cancer cell, is the ability to spread and invade, which we call metastasis. So I don't think I have time to go into your question in great detail here, David, but these two phenomena are linked, immortality and what we call programmed cell death. Is that okay?
Yes, sir.
Thank you.
Thank you.
Okay, so here are some comments on cancer characteristics. Most of the characteristics shown in the previous slide are due to mutations in key genes that make proteins that regulate, for example, cell growth. Cell growth is abnormal in cancer. Cancer cells grow rapidly and they grow independently of and don't respond to various hormones or growth factors that normal cells do. So generally, many cancer cells show rapid, uncontrolled growth. Very often this is called autonomous, A-U-T-O-N-O-M-O-U-S. That is, the cancer cell does not respond to normal signals that normal cells would. Okay? So there are what we call in the body cell signaling pathways, and I'll talk about them briefly. Okay? I mentioned apoptosis, programmed cell death. In terms of metastasis, a very important phenomenon that I'll talk about briefly is that normal cells stick to each other. Okay? They're quite adhesive, whereas cancer cells tend to be less adhesive. They show less adhesion to each other, so that a cancer cell can break off from a cancer and then move through the tissue and get to a blood vessel or get to a lymphatic vessel and then spread to the rest of the body. And another important aspect is there's a whole complex process involving a variety of enzymes and reactions called DNA repair, okay? And if a chemical interacts with DNA in a normal cell, that can sometimes be repaired. Enzymes can hone in on the DNA, chop out the abnormal part, and repair it. But if you have mutations in any of the enzymes that carry out DNA repair, if they're changed and don't do their job, then the DNA will not be repaired, and that can be an important contributor to cancer, because the DNA is not repaired, the mutations are not repaired, the genes are permanently altered, and they contribute to the characteristics the abnormal characteristics of cancer cells that I mentioned in the previous slide. Okay, so two types of genes have been of great importance in recent cancer research. Okay, they are oncogenes and tumor suppressor genes. They emerged about 1980 or so. So a lot of people are skeptical about the progress that's been made at cancer, but actually at the basic level, a tremendous progress has been made in understanding how cells become cancerous. And the concept of oncogenes and tumor suppressor genes were key concepts in our recent understanding of the behavior of cancer cells. An oncogene then, oncos from the word a tumor, okay, these are genes that are involved in promoting the growth of cancer cells. They were first recognized in certain tumor viruses. As you may know, there are some viruses that can cause tumors, and they've been extensively studied by cancer researchers and virologists, okay? And then it was found out that normal cells contain certain genes that were very similar to these oncogenes, and these are called proto-oncogenes. That means they were precursors. Proto means precursor, or occurring prior to the change of the gene making it an oncogene. So that if these proto-oncogenes are mutated, they produce specific proteins that drive cell growth. And that, of course, is one of the most important features of cancer, continued unregulated cell growth. So that these mutated genes, which are derived from normal cellular genes, which are called proto-oncogenes, the mutated genes are called oncogenes. Okay? Now, tumor suppressor genes are, again, very important in cancer. Normal cells contain genes that produce proteins that suppress cell growth. Okay? So these are called tumor suppressor genes. If they are mutated, if these genes are mutated, and I pointed out earlier that mutation is a key event in cancer, okay? Changes are critical genes, not necessarily all the genes of a cell, but certain genes are involved in growth, certain genes are involved in determining the surfaces of cancer cells, etc. But if the tumor suppressor genes, if they mutate, then the proteins that they produce no longer suppress cell growth, and I think you could see intuitively that that is going to contribute to rapid cell growth. So you have oncogenes, the previous slide, which the products of the oncogenes drive cell growth, and conversely, if you lose a tumor suppressor gene, it's normally inhibiting cell growth, so if you lose it, it no longer inhibits cell growth, and that contributes to rapid cell growth. And a whole variety of oncogenes and tumor suppressor genes have been discovered, and various workers all through the world are studying one particular oncogene or a variety of oncogenes. Others are studying tumor suppressor genes, etc. So conceptually now we have the basis of an understanding of cancer, that certain genes contribute to cell growth, whether it be oncogenes driving cell growth or tumor suppressor genes. If they're knocked out, they no longer suppress cell growth, and that would contribute to cancer. Okay? So cell signaling I've talked about. What do we mean by that? I don't see my pointer working here too well. The mouse, maybe, where am I? Okay, I'm trying to get the mouse to work, but here we are. Here's the surface of a cell. As some of you know, on the surfaces of cells, there are proteins called receptors. This is one receptor, R is receptor. This is a different receptor, this is a different receptor, this is a different receptor. So in the surfaces of cells, there's a variety of receptors for different hormones, for different growth factors, for drugs, for nutrients, etc. Okay? So many of these receptors on the cell surface are in fact what we call glycoproteins. These are proteins that contain sugar chains, okay? But let's say this is a signal, okay? Let's say this is a cell that responds to insulin. Then insulin is a signal. This is the receptor for insulin. When insulin interacts with the receptor, binds to the receptor, that changes the receptor, and that sends off chemical molecules, without going into the details, that migrate into the nucleus, and they specifically affect the activity of certain genes. They turn on certain genes, or they turn off certain genes. So we have a whole variety of different receptors with different pathways. The pathway whereby this receptor influences genes is different from this one, and this one's different from R2 and R1. So there's a whole variety of signaling pathways which basically eventually turn on and turn off the activity of certain genes. And we're concerned here mostly with genes that are affecting cell growth and some of the characteristics of cancer cells. So this is what we mean by cell signaling. There's a variety of signaling pathways in which a molecule, a signaling molecule, a hormone, or a growth factor, or other molecules interacts with the receptor, sends in chemicals that turn on or off certain key genes in the nucleus. Okay? Now, so here's some comments in signaling. Each signaling pathway, and there may be 20, 30 more, many more signaling pathways in a cell, has different components. It's quite complex, but that's the way it is. Proteins, enzymes, other molecules, and they regulate different cell processes, such as programmed cell death, I mentioned that earlier, DNA repair, the ability to repair DNA if it's damaged. If you lose that ability, then a DNA is going to be permanently changed or mutated. Signals regulate cell division and growth. Other signals affect the way cells stick together, and others affect the ability of a cell to invade the tissues and to spread elsewhere in the body. For example, a recent study on pancreatic cancer showed that 12 important signaling pathways have been recognized in human pancreatic cancer as altered. As you may know, human pancreatic cancer is one of the most serious cancers. It's the survival figures for pancreatic cancer are not that impressive. So this is a paper that you could access if you were interested. This came out very recently in the prestigious journal Science, September 26. This is a study on pancreatic cancer. 24 human pancreatic tumors were analyzed. Amazingly, the technology is there now. 20,000 plus genes were sequenced and studied, looking for changes in the genes. On average, human pancreatic cancers contain an average of 63 genetic alterations. This is what this study from Johns Hopkins University showed. The alterations, in fact, define 12 of these signaling pathways that I just talked about, and they were altered, these 12, some of them, the signaling pathways were altered in all the tumors, and others were altered in a certain percentage of the tumors. And the authors believe that faulty regulation due to mutations of these signaling pathways can explain the major features of pancreatic cancers. That is, the rapid growth, the spread, and many other properties that I'm not detailing. So this is a very recent study, and the same issue of Science contained a similar article on one particular type of human brain tumor. So the technology there is now to study the products of 20,000 genes is a lot of work, but there's now instrumentation available at many of the big laboratories throughout the world used to study and acquire knowledge, basic knowledge of cancer. Okay? So here's some other characteristics of cancer cells. A very important feature is that they show changes in cell surface molecules, for example, glycoproteins and other molecules involved in the way cells stick together. And that makes sense because we know that cancer cells are less adhesive, and the way cells adhere to each other is by sticking to each other. So people are looking for biochemical changes in the molecules on the surfaces of cancer cells, comparing them with normal cells, okay? Trying to explain why cancer cells don't stick together, and this facilitates the phenomenon of metastasis. Now, another important aspect of cancer cells is that they show an abnormal number of chromosomes, okay? When the pathologist gets a specimen of cancer, if you have an operation and it is cancer, the pathologist in the hospital looks at the material under the microscope, and if it's quite aggressive cancer, you see abnormal number of chromosomes. And in the Scientific American article that I mentioned to you, this phenomenon, which is called aneuploidy, is under a lot of study. How do you generate an abnormal number of chromosomes in cancer cells? As I mentioned, I've lost the mouse again is not pointing to, yeah, here we are. Abnormalities of DNA repair mechanisms. Okay, if any of the enzymes involved in DNA repair are altered by mutation, okay, that area of damaged DNA, if it be this area here, is not going to be repaired, and if it contains a critical gene, that will change the cell or help to change the cell to becoming a cancer cell. There are various abnormalities of the metabolism of cancer cells, which I'm not going to discuss here tonight, but these have been much studied. The enzyme patterns of cancer cells often show quite major differences from those of normal cells, and this results in abnormal metabolism. An interesting and useful phenomenon is that many cancer cells secrete various proteins into the surrounding tissue and into the blood, and you can, some of the tests for cancer involve measuring these various proteins in the blood, and these are called tumor biomarkers, okay? There are certain proteins in the blood that increase in cancer. They can be used to help the diagnosis of cancer, and they can be used to follow whether cancer is being successfully treated. If the amount of the biomarker in the blood drops, that suggests that the tumor is being killed. On the other hand, if eventually the protein, the biomarker, reappears in the blood after an interval, that could suggest a recurrence of the cancer. Okay? Now, another whole area that, as many of you know, there's great interest in stem cells, and there's a whole literature now coming up on the involvement of stem cells in cancer. Basically, a stem cell here is shown here, and they have stem cells. When they divide, they reproduce themselves to make one copy of themselves, original cell, and the other cell that is made when the stem cell divides leads to differentiated cell, okay? So this is called renewing the stem cell supply. On the other hand, this offspring can become a differentiated cell. By that we mean that it can become a cell in the brain, in the muscle, in the kidney, many, many different cell types. So there's quite a large literature now on stem cells, and the article in Scientific American, there's an article in that Scientific American issue on the involvement of stem cells in cancer. So a stem cell is a primitive cell that has the capacity for self-renewal and also for differentiation into a variety of mature cells. Okay? And evidence is accumulated that certain stem cells possess the capacity to become malignant, to become cancerous. And this could be very important and maybe one explanation of recurrences of cancer. If these potentially malignant stem cells are not killed off, the cancer can recur. Even if you kill the cancer cells, if there are malignant stem cells, that can pose a problem because they can divide and make more malignant cells. So this is becoming quite an important area in current cancer research. Okay? Now in this slide, I'm talking a little bit about cancer cells adhere less tightly to each other and can metastasize, they can spread. So here we see normal cells. They adhere tightly to each other, probably through proteins on the cell surface and other molecules as well. Okay? In cancer cells, they, as I tried to indicate here, they adhere much less tightly. So this cell here can bud off from a cancer. It can migrate through the tissues, and it can get to a lymphatic or into a blood vessel, or as I mentioned earlier, in the abdomen. It can spread across the abdomen, and eventually it can take off to other organs in the body where the cancer cells should not be, and they can grow there and cause serious problems. Okay? Now, so one cause of lessened adhesion of cancer cells is indicated here. Here we have two cells, okay, normal cells, and there's been a lot of research being done on how the cells stick together. Like liver cells stick to liver cells, brain cells stick to brain cells, muscle cells to muscle cells. What is the mechanism of how cells stick together? And this is not just of academic interest because, as I've indicated, cancer cells do not stick together as tightly as normal cells. So it's been found out that there are proteins on the surfaces of normal cells called cell adhesion molecules, so that this cell sticks to this cell in part through the interaction of these two cell adhesion molecules. This is one cell adhesion molecule, this is another, they're interacting. Okay? So if you change the structure of these cell adhesion molecules, if mutations in genes which encode or make the cell adhesion molecules, mutations in these genes can be involved in the decreased cell adhesion found in cancer cells. Now that's only one aspect trying to explain the differences in the cell surfaces of cancer cells. This is a big area of research studying the chemical properties of normal cells and cancer cells, comparing them to see what the chemical differences are, and will that help to explain the phenomenon of metastasis. Okay, here we see a diagram. Again, I seem to have lost the mouse here. Here we are. Okay. Sorry?
Anytime you right-click or left-click, it might appear after that.
Okay, thank you, David. Okay, here we see a patient who had what we call a cancer of the colon. This is the transverse colon here. So that's the primary tumor, and metastasis can occur very often in the case of colon cancer to the liver, okay, to the lungs, okay, to the brain, of course, that is obviously serious, or to the skeleton. So these indicate metastasis. These are called secondary tumors then because they're produced by metastasis, whereas in this case, the primary tumor was in the colon. If the primary tumor was in the lung, then we could have malignant cells from the lung going to the liver, going to the bone or the brain, or if the primary tumor was in the breast, the cells can go to the lung or the brain or the bone, and these are obviously, that's a serious situation for the patient. Okay? Now, so here we, sorry?
And the spread is it based on the lymphatic or the venous drainage? Is that like a pattern to which kinds of cancers would spread to which parts of the body?
Yeah, that's a good question. In the prostate, for example, they will often spread in the venous supply in the pelvis to the skeleton. The exact factors that govern metastasis, why some cells go to the lung and go to the liver, there's a lot of study being done in that, why some go to the brain. Another interesting phenomenon is that muscle is about 40 or 50% of the weight of the body, and yet metastasis to muscle are very, very unusual. Tumors metastasize like the colon to the liver or the lung or to the skeleton. So many investigators are using mice models, or trying to also look in specimens of human tumors to try to explain the specific patterns of metastasis that are found. Why, as I said, some tumors go to the lung, why some prefer the brain, etc. So that's a very important issue that we don't have time to discuss here tonight. But again, if you're interested, you could access metastasis, and there are many papers appearing studying the phenomena of metastasis. So at a more practical or everyday level, if you've had an operation for, say, cancer of the colon, the tumor is removed along with the adjacent tissue, a border of normal tissue to make sure you get rid of all the cancer. Then the material from the tumor is examined microscopically and also visually by the pathologist, okay? And the pathologist can do this quickly in what they call a frozen section, in which they look at the tissue during the operation, but they also take tissue and it's worked up in the lab to get a better specimens, and that may take a few days after the operation, even a week sometimes, to get the final report from the pathologist. So the pathologist looks at the microscopic pattern of the tumor, and also visually, if it's a specimen of colon, they describe the size of the colon. They look to see if the lymph glands that are close to the colon are involved. And of course, if the surgeon finds that the tumor has spread to the liver, then that is noted, etc. Or if X-rays show that it's spread elsewhere. So subsequently, the tumor is graded and staged. Okay, these are the two terms. So what do these terms mean, and what is their significance for a patient? Well, grading tumors, this is actually based on how similar the tumor cells look to normal. Okay, we call that the degree of differentiation. If the tumor is well differentiated, that means it's very, very similar to normal. Okay, and that's relatively generally good. Okay, because it means the tumor has a lot of, if you want to call it inverted commas, normal-like features. Okay, so they look at how similar the tumor cells are to normal cells, and they look to see the number of cells that are dividing. That is called mitosis, the phenomenon of cell division. And a pathologist, when he looks at the stained section, they will often count the number of cells that are dividing. In normal tissue, very few cells will be dividing. In tumor tissue, a lot more cells will be dividing because that's a major feature of cancer cells, that they divide frequently and rapidly. And this can be assessed by microscopic examination. And then they grade the features 1 to 4. So that grade 4 is the most serious because the cells are least like normal cells, and thus generally most malignant, okay? But overall, grading is not quite as reliable as a procedure as the next procedure, which is called staging. So grading is really looking at the microscopic features of the cancer cells, and a relatively low grade is 1, which is generally good or better, and 4 would be a pretty serious, that would indicate cells that are rapidly growing and have lost many of the features of normal cells. So staging tumors then is based on the size of the tumor, the extent of spread to the local lymph glands, okay? We know that many of us know that if you have cancer of the breast, that an important feature are the local lymph nodes involved, okay? And the presence or absence of metastasis. If it's the colon, if there's metastasis in the liver or the lung or elsewhere, then that is a more serious situation. So the TNM system is one system. There are other systems that are used to stage tumors, but T is the size of the tumor, N is the number of local lymph nodes involved, and M is metastasis. So a tumor that would be T1, M1, M0 would be a small tumor, T the size of the tumor, N would mean only a small number of lymph nodes are involved, and there are no metastases. So that's good, okay? So it's a small tumor, it's only in a few lymph glands, and there's no metastases. The best, obviously, would be T0, N0, M0, T0, N0, M0. So what uses is staging then? Will assist in estimating the outlook. For example, T4, N4, M4 would be a pretty serious situation, okay? Whereas T1, we have a small tumor, there's no tumor in the lymph nodes, there's no metastases, that would be relatively good. And it's also useful in monitoring following treatment. For example, the TNM staging will decrease or lower if the cancer shrinks and the local lymph glands or lymph nodes, these terms, gland and node, are interchangeable, are cleared of tumors. So this staging of cancer is an important procedure by the clinicians and the pathologist, okay? So I'm only going to mention superficially some types of treatment. I'm not going to go into natural therapies. I know that a lot of people out there are interested. That might be the subject some other time of another seminar. But the standard medical treatments are mainly, as you know, chemotherapy, okay, radiation, different types of radiation, and immunotherapy using aspects of the immune system, antibodies or various immune cells. So chemotherapy, as probably some of you know, is often a cocktail. That means a mixture of 3 or 4 different drugs. And the reason for that is if you use one drug, the cancer cell can become resistant to the drug, okay, by mechanisms that are being studied, because that's a very important phenomenon, how cancer cells become resistant to chemotherapy. I won't go into that right now. But if you give a mixture of, say, 3 or 4 different chemotherapeutic agents, then the cancer cell is less likely to become resistant to that mixture, okay? So a few comments on treatment then is that many chemotherapy drugs act to damage DNA and thus kill the cancer cells. That's not the only way in which chemotherapeutic drugs act, but it's a way in which many of them act. They attack the DNA and damage it, and that will result in killing of the cancer cells. But obviously, that can also damage normal cells, okay? Because although the cancer cell may take up more of the chemotherapeutic drug, normal cells will take them up to a certain extent, and their DNA is going to be damaged. So chemotherapy, one problem then is lack of specificity, okay? It would be nice to have a chemotherapeutic drug that only entered and only damaged the cancer cells, okay? Now, another problem, as I said, is development of resistance, okay? The cancer cell adapts to cope with the drug. For example, some cancer cells that are resistant actually pump the chemotherapeutic drug out of the cell into the environment, and to get rid of the drug, by mechanisms that are being studied, the cancer cell adapts. It gets a protein on its surface that can pump the chemotherapeutic drug out of the cell, and that decreases its damaging effect on the cancer cell. Now, at least in theory, immune approaches may offer greater specificity, because if you find an abnormal molecule on the surface of a cancer cell, you can perhaps make an antibody to that one specific molecule which is abnormal and hopefully not abnormal in cancer cells, and then you can use that antibody to attack and damage the cancer cell. So there's great hope for immunotherapy, although as yet it's still very much a developing area, immunotherapy, okay? But people are hopeful that as the immune system is studied in more and more detail, the various cells in the immune system, some of you may have heard of natural killer cells and other cells that can attack cancer cells, antibodies, which are proteins, they can latch on to specific abnormal molecules in cancer cells. You can sometimes tie a toxic drug onto the antibody, and that will damage, the antibody will be taken up by the cancer cell, and the toxic drug or some other agent can be released and help to kill the cancer cell, okay? So now here's a drug that some of you may have heard of, okay? This is a case of, in about 20% of breast cancers, there is an increase of this particular receptor, okay? This is a receptor, a human epidermal growth factor. That's a growth factor, and if this receptor increases on the cell surface because of changes in the gene, okay, the gene is affected in the cancer, more of the receptor is made, and if there's more of the receptor, then a natural factor called the ligand, where the growth factor can sit on the receptor, and it can stimulate the receptor and send out messages that make the cell grow or proliferate. And this would be, for example, 20% of human breast cancers have this increased amount of this particular receptor, which is called HER2. Okay, that's it here. So normally, Herceptin is not here, so the growth factor sits right on the receptor. But the Herceptin, it's also got this name here, trastuzumab, okay? What it does is it is an antibody, okay? The last two letters indicate antibody, and it sits on the receptor and blocks the receptor and prevents the growth factor stimulating the receptor. And this can eventually lead to death of these cancer cells. So this is an example of a drug that is actually an antibody. It's part of immunotherapy. But what it's doing is it sits on the receptor and prevents the factor that would normally sit on the receptor, causing stimulation of growth. That is inhibited or cut out by Herceptin. Okay. Now, eventually, resistance to Herceptin might occur, but there's a whole variety of these different antibodies being developed, and they appear to be a promising area of cancer research. Okay? So, Herceptin, I'm sure some of you at least may have heard of it. HER2 is a receptor for this particular growth factor, which stimulates the growth of cells by sitting on the receptor. 20% of breast cancer cells, human breast cancer cells, overproduce this receptor. Overproduction of the receptor stimulates the breast cancer cells to multiply, and Herceptin then is an antibody to the receptor. It interacts with the receptor and blocks its action. So that's an example of immunotherapy that is being widely used in North America and elsewhere for certain patients with breast cancer. Okay? So immunotherapy then, the immune system can recognize many molecules very specifically, for example, altered proteins. So in theory, if there's a protein altered in a cancer cell, you may be able to make an antibody to it. If you purify the abnormal protein, inject it into an animal, or there are other techniques now for making antibodies, you can raise antibodies to that altered protein. Then you can use these antibodies to try and attack the cancer cell. Okay? So specificity for such molecules in cancer cells could cut down the side effects of therapy. Okay? The antibody would be relatively specific for the cancer cell. And various approaches are under study in this area of immunotherapy. Antibodies to cancer-specific molecules are being studied. Some of them can kill cells. Attempts to increase the immune reactions of immune cells from cancer patients. You can take out the immune cells in the blood, put them into tissue culture, try to increase their immune ability, and put them back into the cancer patient and see whether hopefully that can affect the cancer. And vaccines to tumor cells, to specific components of tumor cells, are also being investigated. So this is, again, an active area of research at the present time. And two other treatments that are being actively investigated, and I should say that if you are interested in this area, that Scientific American issue covers many of the topics that I've been talking about tonight, okay? Two other treatments, drugs that attack the blood supply of tumors specifically. I mentioned already that abnormal blood supply is a feature of many tumors. If you can cut off the blood supply by certain drugs or other agents, then the tumor cells will die. And there's a lot of research going on in this particular area, which is called angiogenesis. Okay? Then another development is that some pigments, it's been found that certain pigments can be taken up by tumor cells more or less selectively. The tumor cells take up much more than normal, and then once you're taken up, if you then expose the tumor cells to light or to laser, that changes the pigments that have been taken up or the drugs and makes them toxic. And this is called phototherapy, light therapy, in which specific agents are taken up by tumor cells. Then you shine some type of light on the tumor cell, and this can prove toxic to the tumor cell if it has taken up much more than the normal cell has. Okay? Now, at a practical level, I'm sure there's nothing here that is new to most of you listeners, okay? In terms of prevention, then, nutrition is always important in almost every health situation, okay? Taking ample fruits and vegetables and appropriate supplements. Smoking, of course, is a no-no. Minimal alcohol intake, maybe a glass or two of red wine. There's a lot of interest in red wine in the molecule that it contains resveratrol that may have different properties that are being studied in many labs, okay? But moderate or minimal alcohol, because alcohol has been shown to increase the mutation power of certain chemicals. If you're working with chemicals, then you want to know as much as possible. If you're in a factory industrial job, then you want to know as much as possible about the agents that you may be exposed to. It's a good idea to know your family history, and either our gene tests for a few cancers. I mentioned earlier the cancer susceptibility genes for breast and, for example, ovarian cancer, and some women are availing themselves of that if there's a history in the family of breast cancer or ovarian cancer, although I should say that only a relatively small number of women have these abnormal genes. It's quite a small figure. Then, of course, it's important to get regular checkups, okay? And go early for a checkup if you have any suspicious signs or symptoms. We know these signs, probably bleeding, skin growth, chronic cough, change of bowel movements, fatigue, unexplained weight loss, fever, persistent pain, etc. Most of you are familiar with these, but most of us stall and put off going to the right healthcare professionals. So that overall, early detection is a very important feature in successful treatment of cancer. If you can go early when it's T1, N0, M0, then your chances of successfully going through the ordeal are just infinitely greater than if the tumor has spread to lymph nodes or particularly to other organs in the body. The statistics out there for different cancers at different stages, but I'm not going to go into that at this stage. So in summary, let me see. Despite some people's reservations, I think much has been learned about the behavior of cancer cells. At a basic level, people interested in cancer, now we have a pretty good picture of what can make a cell become cancerous. Oncogenes, tumor suppressor genes, other genes, etc., DNA repair, without repeating what I said earlier. And new genomic approaches to learning more about the abnormalities of cancer cells are very promising. For example, the study in pancreatic cancer where 20,000 genes were sequenced by appropriate technology. And these kind of studies are going to go on in every type of human cancer, brain, every organ. And this will give us, I think, a lot of information and hopefully point out the products of cancer genes that may be susceptible to drug or nutritional or other approaches, okay? So new and better treatments based on research are being introduced. I think it's important, it's easy for me to say, and I probably haven't adopted a particularly healthy lifestyle myself, but I don't smoke, and a long time ago I did smoke, but I cut it out, and I don't take too much alcohol. I should exercise more, etc. But I think the single most important factor is still early diagnosis. If you're concerned about something, do not delay if you're a sign or symptom that may indicate cancer. So I've tried to give you a kind of overview, I think, that incorporates at a very general level how many doctors and other people in healthcare think about cancer these days. Now, I know that not everyone will share the same views as I have, but I think I would call them probably the standard medical approach. But I do believe that research is very, very important, and it's through research, the study by basic scientists, not physicians necessarily, but people in cancer research, studying genes, etc., that they have given us very important insights into the nature of cancer. And if you have any questions, I am happy to, if you email them to me, I can try and answer them. I may not be successful in quite a number of cases. Thank you for listening, and for David probably has some remarks.
Yes, sir. Well, thank you very, very much, Dr. Murray. Actually, there have been a lot of questions being typed in while you were speaking. We'll just pick a few, and based on how many you can answer, we'll just work on it that way. Folks, real quick, I just wanted to add, before I start asking the questions, there was this interesting study that was done a couple of years ago in which they I think they were breast cancer patients, and what they wanted to find out was how social support, having a good family around the patient, how that helped in the prognosis of the treatment. And here's what they found out. They found out over and over again that good social support helped to double the people who had good social support had double the length of life. Double the what's the word now?
Expectancy.
Life expectancy. Thank you, sir. Life expectancy than those who did not have good social support. So that is another thing. I mean, if you have friends or family or you yourself are going through cancer, it'd be a tremendous help to have that kind of support along with.
Plus attitude. Attitude, I think, is very important.
Yes, sir.
Absolutely. You know, a positive attitude is very important.
But that's the problem, because when people get cancer, they're so full of fear and they just break down, and that translates into their immune system, and their bodies just shut down. And I experience that a lot with my cancer patients too. So
I think that's where support groups are very important, where people talk to other people who have hopefully undergone successful treatment. As you say, social, psychological interaction. I didn't cover that here, but these are all very important features of undergoing cancer treatment.
Absolutely, absolutely. All right. Couple of things. Just to reiterate what you just said. So cancer, from what you just said, there are about four main things that characterize cancer. One is that the cells suddenly decide to keep growing and reproducing. It's like the brakes are turned off. Secondly is that cells, they are not suppressed. They're basically the same thing. The brakes are turned off, accelerating, and their growth and reproduction accelerates. The third is that their signaling is now warped, and so they don't respond to normal signals and also give abnormal signals to the rest of the body.
Yep.
And number four is that they break away in a process called metastasis. So does that summarize what you just said?
I would say that's good. Sometimes the changes may not be that sudden. The changes may evolve over a period of time. So sudden would be the only thing I might debate with you. That these changes, the genetic changes, may take time to accumulate the variety of mutations that you find in a cancer cell. And also, I should have mentioned maybe that there's evidence that cancer cells are being normally produced in our bodies, but in small amounts that the immune system can take care of. But if you're immunosuppressed, if you have a disease that suppresses your immune system, or if you're taking drugs that suppress your immune system, sometimes the cancer will appear because the immune system is not as active as it should be.
Right, right. And that brings up a point you also made when you said our repair mechanisms are not keeping up with the damage to the DNA, and so that overcomes and overrides and eventually leads to cancer, right?
Yeah, I think the DNA repair mechanisms, there's 6 or 7 different mechanisms of repairing DNA. There are whole books on DNA repair, okay, how DNA is repaired. And if you have mutations in a key DNA repair enzyme, then the DNA is not going to be repaired. And that can certainly, there are a few genetic diseases in which that is occurring, mutations of DNA repair enzymes. And these, there's one particular condition called xeroderma pigmentosum. Okay, it's dry pigmented skin, and children can develop cancers because the DNA repair enzyme is not acting properly, and they get mutations which are not repaired.
Someone has a question about malignant potential. I'm not familiar with the term. Are you?
Well, malignant potential to me would be some cancers are, one aspect of it would be that some cancers are a lot more aggressive than others, and some are relatively slow growing, okay? You know, you could have a cancer of the colon in one person that would be relatively slow growing, whereas in another one you could have a cancer of the colon that is rapidly growing, gets out into the lymph nodes, metastasizes. So that's one aspect of malignant potential. I don't know if that's what the listener had in mind.
Mm-hmm. Well, yeah, nothing about metastasis. I understand over 90% of all deaths in cancer is actually due to that process.
Yeah, metastasis is, you know, as I said before, if you're in stage 4, there are survival figures for when the tumor is out of the original site and into the lung or elsewhere, and the figures are a lot more gloomy depending on the amount of spread, amount of metastasis. But
right.
Hopefully, as the phenomenon of metastasis is studied more and more, you know, maybe there will be ways of preventing metastasis. If you could convert a malignant tumor to a benign tumor, I don't know if that will ever be possible, but maybe, who knows, with the rate of advances, that would be fantastic. You know, if you could somehow stop metastasis.
Right.
You still have to deal with the tumor, but if it's locally, if it's just in the colon, then a surgical operation can get rid of it.
Right. Yeah. Solving metastasis really is the key to helping cancer patients. Here's a question on leukemia, and
yeah.
I saw a movie yesterday about, it's a true life story about this kid who was cut down in the prime of his life at 23. He won the Heisman football trophy, and I think in the 1960s. A talented guy, but he came down with leukemia. And anyway, someone says ask Dr. Murray if his comments about cancer also apply to leukemia when he's talking about cells and glycoforms and glycoconjugate structures, etc.
Yeah, leukemia, some of the comments, you know, blood cells are normally circulating, so in a sense they're not metastasizing because they're normally in the circulation, whereas colon cells are not normally in the circulation. But certainly the cells of acute leukemia, whether it be acute lymphatic or acute myeloid leukemia, they're very rapidly growing. So that's an important feature. They're very rapidly growing and they're relatively de-differentiated. Okay? Now, if you have chronic leukemia, sometimes chronic lymphatic leukemia, for example, can go on for 20 years or more. It can be quite a slow, indolent disease. So acute leukemia generally can advance very rapidly, but particularly in childhood leukemias, there's been tremendous successes with chemotherapy and supporting therapy. For example, St. Jude's Hospital in Memphis, Tennessee, they have excellent figures for many of the childhood leukemias. But to go back to the question, some of the characteristics of cancer cells would be in leukemia cells, rapid growth, etc., metabolic changes in the cells, and a variety of mutations in the cells, etc. But you don't normally talk of leukemic cells as metastasizing because, in a sense, they are already in the blood. Of course, they can settle out in the brain or they can settle out elsewhere. Whether you call that metastasis or just blood spread is maybe.
Right.
Yeah.
Right.
That's a good question.
Yeah, and the trouble with leukemia, again, is that because it's in the blood, it's actually a cancer of your white blood cells in some cases.
Yep.
And the white blood cells are the immune system. So it's tough for the immune system to fight against something that is when it's a part of the problem itself. So that's
right, yeah. That's why these people, patients with leukemia, are susceptible to infections. They can be susceptible to bleeding because the platelets get squeezed out. These are the cells that are normally involved in blood coagulation, and then also the heavy chemotherapy can produce toxic effects too. But there has been tremendous progress made, particularly in the treatment of childhood leukemias.
Right, right. Is it true that cancer cannot exist in an alkaline environment? If so, why isn't more attention given to a person's blood pH?
Yeah, I'm not a big fan. I know there are people, I think there's a clinic in Rome where people go to, and then there's also, I think, the other clinics where they talk about fungus as being a cause of cancer. I'm not absolutely convinced about the alkalinity of tumors. Certainly, tumors often are in a kind of acidic medium. They tend to grow rapidly, and they often have high rate of breaking down sugars that produce acids. So, from my perspective, I'm not that impressed about trying to change the pH of cancer cells because the body has so many buffer systems and tries to maintain the pH within a very narrow limit. In fact, if your pH drops below 7, then you're very, very, very sick. So there's not a lot of room to change pH. But
right.
That's just there's probably other people out there who may be big proponents of pH, treating cancers by altered pH.
Right.
pH is a term for acidity or alkalinity, pH.
Right. I have a question from Dr. Steve Summey. I know you're familiar with him.
Yes. How are you? I guess he's in Colorado, I think.
Yes, he is. He is in Longmont, Colorado. Dr. Murray, I know you may be limited in this area due to regulatory restrictions, but can you comment on the importance of prevention of cancer initiation, progression, and metastasis with supplemental antioxidants like glutathione, catalase, and superoxide dismutase in DNA repair and cellular oxidative protection with chemotherapy? Wow. What about the phytochemicals in the cruciferous vegetables in the same question above? Thanks, Dr. Steve.
I think Dr. Summey might do a better job of answering that question than I would.
Could you repeat that, David? Could you repeat that question, please?
Okay.
It's almost the Encyclopedia Britannica question.
And well, okay. Well, would you like to comment on the?
Well, I believe in taking antioxidants in certain vegetables in prevention of cancer. I'm not so sure what success they have in the actual therapy of cancer. I mean, all these superoxide dismutase is a very important cellular enzyme. Glutathione is the major chemical in our cells that regulates the oxidation reduction potential, reducing atmosphere of cell. So they're all very important molecules. I don't know if the question is giving them individually to people, are they effective cancer agents? In many cases, they would not get into cells. Superoxide dismutase wouldn't be taken up we don't know that it would be taken up by the cancer cells. So they're very important chemicals in terms of the normal biochemistry of the cell, and they may also be very important in understanding the biochemistry of the cancer cell. But I can't comment on trials. I don't have the information at my fingertips as to how successfully any of these agents have been used in cancer therapy. Dr. Summey may know. For myself, I would like to see the results of a clinical trial of, say, 50 or 100 people and 50% got the agent and 50% got a placebo, and how that would affect, say, survival. That would be, but I don't know of a big literature regarding the agents that Dr. Summey mentioned, but he may, in fact, know, as I said, a lot more about that than I do.
Dr. Summey, are you unmuted?
Yeah, can you hear me?
Yeah, we can hear you.
Hey, thanks, Doc. Appreciate the effort. No, I think that in some of my studies with some of the agencies, the researchers, I don't think there's too much disagreement in the fact that these agents, all these chemotherapeutic phytonutrients, have a major role in prevention. I think there's a lot of, I can't right off the bat come up with the particular studies, but I think that a lot of the American Cancer Society and a lot of doctors believe that these cruciferous, these chemicals that are in these vegetables have a major role in prevention.
Yeah, I would go along with that.
Yeah, so no, I wasn't referring from a treatment after the fact standpoint. I think that that's kind of where we're trying to attack this from a health broad-based point of view from a large population. Is let's try and get ahead of this problem.
Yeah, I think every week I'm reading about how good broccoli is, and I think, as I did mention in the slide before the summary, fruits and vegetables, and I mean, a lot of us have been abusing ourselves for many years. I know I have. When I was a student, every day for 6 years I had pie and chips, pie and French fry. I can't think of a worse diet to have had. So I think dietary factors are very important, and more and more attention is going to be paid to that. So I go along with Dr. Summey, all these phytonutrients, and I think they have to be scientifically investigated. That's the only point I would make. I like to see, fairly definitive evidence that they are of benefit.
Yeah, I think a lot of the larger research institutions are finally starting to move in this direction. I think they've had a lot of push from groups like, right now what's going on is this raw food. It's a week-long program with guest speakers that are happening every night, and so we know that getting the organic or the raw foods and getting the live enzymes and getting all these phytochemicals that help give us that protection that's so important in repairing the damage, because as it gets progressing down the road, then like you mentioned, you get beyond the point of no return, and it really becomes very difficult to treat once it gets to that stage.
Absolutely, I subscribe absolutely to your comments.
Thanks, Doc.
Yeah, look after yourself.
All right, thanks.
Thanks, Dr.
Bye.
And actually, Dr. Reg was on the call too. Dr. Reg McDaniel.
Oh dear, I'm probably in trouble. I hope he wasn't taking notes.
Well, actually, he said you and he actually did a seminar together in Calgary. When was that?
That's right. Yeah, I think Dr. Summy may have been there. I'm not sure. Yeah. Anyway, best wishes to Dr. Reg.
Yeah, he
and to his brother.
Okay, we'll let him know.
Okay.
Somebody asked about hormonal therapy. I don't think you mentioned that in your notes.
Yeah, well, to a certain extent, yeah, in terms of receptors, okay. I didn't want to go into. I'm trying to remember the name of the drug that was used for breast cancer, but again, it was based on the fact that it inhibited a hormone receptor in the breast, and certainly, whether, for example, in breast, whether a tumor is responsive or not to hormones is a very important factor in the treatment. Tamoxifen? I'm thinking of tamoxifen, okay, that it was a hormone that was designed to, again, inhibit the effects of hormones on receptors in the breast. So certainly hormones are very important factor. I probably should have paid more attention to them, but I was kind of referring to them a little bit under receptors, the hormones, but I should have done more on that.
Right. Well, there's a couple more questions. With the coming of certain new drugs coupled with glycobiology, what do you see on the horizon that will come forward in the next 3 years?
There's been a lot of talk about drugs based on sugar-based therapies, okay? So far as I know, and I'm not quite aware why, I don't think too many successful ones have been brought to the market. But in theory, you can use sugars to direct molecules, for example, to specific receptors. And then if you have a drug connected to the sugar chain, that could be taken up by the cancer cell and exert a toxic effect. So I do believe that sugar changes, for example, on the surfaces of cancer cells are very important in helping to explain. They're one facet of helping to explain metastasis. Metastasis is very complex because the immune system's involved, the movement of cells through tissues, the adhesion of cells. There's a lot of literature. If any of the readers chose to access in PubMed sugar-containing molecules and metastasis, there's a huge body of work in that area. So far, there's not been a lot of translation of it into successful therapy as yet, but my feeling would be that as knowledge of cell surface molecules, particularly glycoproteins, glycolipids, and other sugar-containing molecules, that should give us new agents that will help in the treatment of cancer. But right now, I'm a bit pressed to mention specific molecules. Okay.
Right.
Well, I'm not venturing into the field of nutrition at this stage.
We understand.
I think I told you before that maybe you could give a seminar on the future on nutrition and cancer, or we could talk about it.
Okay. Okay, well, again, you really spent a lot of time on this, and I think we're going to have to wrap it up now, but I just want to thank you again, Dr. Murray. And folks don't know this, but many times when I'm doing a webinar, I'm preparing for a webinar, or I'm going to teach one, I consult with Dr. Murray to if I'm not sure about this issue or that issue. So he's really the one who makes me look smart when I speak on webinars myself. So.
Well, thank you, David. It's always been a pleasure to engage with you. And also, Cherry has been excellent. She was the one that scanned in all my poor illustrations into the PowerPoint slides that I sent her, so I thank her very much.
You're very welcome. Glad to do it.
You don't want to say too many nice things about her, Dr. Murray. You don't want her to say
Yeah.
And thank all the listeners. And if you have any complaints, too, you can email them to us so we can do a better job in the future.
Right. Okay. There you have it, folks. There's the professor who wants to keep on improving. How about that? Okay.
Thank you.
Well, everyone, thank you, and have a good night.
Good night.
Okay. Good night.